My name is Madeleine Garza, and I worked in the Rose Lab during the summer, characterizing the mechanism of cutin biosynthesis in tomato fruit. And so two important concepts to understand in cutin biosynthesis is that one being the function of the cuticle and the other being what the cuticle is exactly. So the cuticle actually has many functions. One of them is regulating water intake and uptake. The other being it protects against UV radiation. Another one being that it prevents organ fusion and is also a self-cleaning. And lastly, it restricts pathogen infection. And all of these are really important as they help the agricultural sector as these combine and limit post-harvest water loss, which thus limits food wastage. And as you can see in this figure, the cuticle is shown right here, and you can see how that protects all the inner layers, such as the cell wall, which is shown by the green, the cytoplasm shown by this dark yellow, and the vacuole by the light blue. There we go. So now that we understand the function of the cuticle, what exactly does the cuticle do? Well, the cuticle lays on the epidermal surface of all land plants and has two lipid constituents, one being an organic compound known as waxes, and the other being a structurally complex of polymers known as cutin. And as you can see in this figure, cutin composes most of the cuticle layer when compared to appendicular wax film and the appendicular wax crystals. So when we're looking for a model for cutin research, what exactly are we looking for? Well, we know that polymerization is extremely complex in cutin. And we also know, as I just said, that the cuticle is mostly made of cutin. So naturally, we want something that has a thick cuticle, thus giving us a lot of room for investigation on cutin. And so that just happened to be the tomato. And as you can see, the tomato has a very thick layer. And so the cuticle is dyed using this oil red O solution, and that is in fact not due to the tomato's red color, but the oil red O solution. And this part is the pericarp here. And the tomato is much thicker than many other models, such as Arabidopsis thaliana, and is just really good for the research that we're doing in the lab. Another reason that the tomato is really good is actually recently the Rose Lab discovered a mutant. And this mutant gives us more insight into the biosynthesis of cutin. As you can see, the mutant is on the lower part and has a 90 to 95% reduction on the amount of cutin that it produces when compared to the M82 wild type that you see on the top. And so we're wondering, well, what exactly is causing this reduction in cutin? And so they mapped the genome, and it was found that a GDSO hydrolase that normally polymerized monomers was missing. And so what that means is usually monomers shown by this arrow will usually be made and polymerized into either linear or branch shapes. However, with that GDSO hydrolase missing, that is known as cutin synthase 1, there's nothing to put together those monomers. Except, as you can see here, strangely enough, there's a very thin residual layer. So what we're trying to figure out is, well, what is making the cutin and the cuticle in that residual layer? And so that's one of the main questions in the lab. And so we looked to see, okay, let's try to find something closely related. So we looked at the phylogenetic tree and found that cutin synthase 2 is in the same clade, and they're also both expressed during tomato development. So you can see, even though they're expressed in development, there are some changes, some differences, as cutin synthase 2 peaks at around 10 dpa, which stands for days post-anthesis, and anthesis is when the flower opens up. And cutin synthase 1 peaks at about 20 dpa and does linger for a little longer. And then I also added some pictures below so that way you can see the rapid growth period here, and then the slow change in color until it gets to the red right. And with this, I hypothesized that a homolog of cus1, which is cutin synthase 2, is responsible for the synthesis of the residual cuticle in the CD1 mutant. And so to characterize and test this hypothesis, I have 3 methods of characterization. 1 being cuticle morphology, 2 being biomechanical properties, and 3 being cutin synthase enzyme activity. So first, I measured cross-sections of many genotypes, specifically 3 genotypes, and I did 2 measurements per genotype. So the first measurement I did was of the cuticle, which is this area, as you can see, and it's the oil red O dyed solution, and from the very top to the very bottom. And the second measurement I did is the upper cuticle, and is just that part, which is the very exterior up until you get to the peg, as is shown by the yellow line. And so the three genotypes that I did these measurements for is wild type, which is shown at the top, known as M82, the mutant, which is known as CD1, and cus2 CRISPR, which is designed using CRISPR. So what are the differences between these genotypes? Well, first, I have the genotype there for you, and then to make it a little easier to understand, I highlighted which gene each one has. So M82, the wild type, has both cutin synthase 1 and cutin synthase 2 gene. And the mutant, CD1, has the cutin synthase 2 gene, but not the cutin synthase 1. And cus2 CRISPR has cutin synthase 1, but not cutin synthase 2. And this one doesn't have cutin synthase 2 due to the fact that a stop codon was added before cutin synthase 2 was read, just so that way we can have a mechanism of comparison when we're comparing M82 and CD1. So just to go over, M82, the wild type, has both of the genes. CD1, the mutant, has only cutin synthase 2, and cus2 CRISPR has only cutin synthase 1. And so after measuring many of these, I was able to compile these and put in some graphs. And as you can see, M82 and cus2 CRISPR are very close in the amount of cuticle that they produce. However, statistically speaking, if we look at M82 and CD1, there's a large significant difference between the two and the amount of cuticle that they produce. And M82, and if you look at the upper cuticle measurements, M82 and cus2 CRISPR are once again close in the amount of cuticle that they produce. And the reason I don't have CD1 for the upper cuticle measurement is that CD1 is so thin that when you look at it under a microscope, it's really difficult to differentiate between the 2 layers, as it appears as just one line. So that's why I only have the entire cuticle for the CD1 mutant. Okay, the next thing I tested are the biomechanical properties, and for this I tested using the Instron machine. And so I cut a piece of the pericarp tissue and I placed this in a pectinase and cellulase solution. And what this solution does is it breaks down the cell walls, so that way we are left with the cuticle only, and there's no disruption when testing the cuticle. And so yes, we're left with the cuticle only. And then we put the cuticle on this Instron machine, and I'll zoom in so you can see it a little bigger. And so that's what it looks like. And the Instron kind of stretches this, and when it stretches it, we're able to come up with some graphs as it gives us the data. So one really important graph is going to be time versus displacement. So you can see that the cus2 CRISPR is shown by red, whereas M82 is shown by wild type, or as shown by black, sorry. And one important thing to note is that they do follow the same curve. However, their displacement level is differentiates among the 2. And lastly, I did cutin synthase enzyme activity, where I performed transient expression in tobacco to test the activities of the cutin synthase 1 and cutin synthase 2 enzymes. So I used the agrobacterium as a natural genetic engineer and then inserted the gene of interest to the agrobacterium. After I did this, I injected this solution into the tobacco leaves. And the reason we use tobacco instead of, let's say, a tomato leaf is because tobacco has an increased level of enzymatic activity, which just gives us more amount of the purified protein as opposed to if we were going to inject it into the tomato leaf. And after this, we let it sit for about a day and then harvest the leaves. So we get the leaves and we crush them up, add a buffer solution, and then we run them through a nickel column, as you can see here. So the nickel column is actually pretty interesting. So what the nickel does, our protein is His tagged, so when the solution runs through the column, the nickel ion attaches to the protein, so that way everything else that is not the protein runs through. And then after that solution runs through, another solution containing imidazole goes through the nickel column, and imidazole displaces the protein, so that way the protein falls through and we're left with only the protein of interest. And after we get the purified protein, we can measure the activity through gel electrophoresis and a Western blot. And so the gel electrophoresis, as you can see here, is shown so that way it proves that we indeed did perform transient expression in tobacco successfully. So you can see cutin synthase 2 is on the left and cutin synthase 1 is on the right, and same order for the Western blot. Now, one really cool thing on the Western blot, as you can see, as this measures the protein concentration, is that there's a really large concentration of cutin synthase 1 as opposed to cutin synthase 2, which I just found really interesting. And so I'm thinking it's because cutin synthase 1 accounts for the larger amount of the cuticle as opposed to cutin synthase 2. And so after I tested my hypothesis, what did I find? Well, in terms of cuticle morphology, I found that there's a significant difference between M82 and CD1. However, there's not a significant difference between M82 and Cus2 CRISPR. And in terms of biomechanical properties, on average, Cus2 CRISPR had a higher displacement level than M82. And lastly, for cutin synthase enzyme activity, I was able to successfully induce protein expression in tobacco, then isolate and measure the protein content through specific antibodies. And I just want to thank everybody in my lab and my mentor who cannot be here, but she really helped me understand everything and gave me really good insight into the lab. And everybody else in my lab, I was very lucky to have a small lab, so I felt that I got really close with the people. And so I just really enjoyed not only the science perspective, but getting to know everybody in the lab. And then a special thank you to Delaney and George who put this together, and of course, Cornell, and especially BTI for having us, as well as NSF and USDA for funding us, so that way I had the great opportunity of coming here. Yes?
With the M82 and then Cus2 CRISPR, how they were virtually identical in one of the first photos that you showed, can you just speculate, like, with the cutin synthase 1 and 2, if they interact, which one goes a lot more than the other?
Yes, so the Cus2 CRISPR did not have cutin synthase 2, but as you saw in the graph, they were strangely close, M82 and Cus2 CRISPR, in the amount of cutin they produced. So what me and my mentor were thinking is that since cutin synthase 1 is such a strong gene, that it in fact overcompensated since cutin synthase 2 was missing, and that's what made the level of Cus2 CRISPR kind of get up there with M82.
So do you have plans to make a double mutant?
Yes, actually, yeah, that was, yes, we have plans to like cross them, and then there's several lineages, and eventually we want to cross it so that way it's recessive for the Cus1 and Cus2, and like supposedly it would be like without, or see, I guess hypothetically what would happen if both of those genes were recessive. So that's kind of, that would take a while, and sadly I can't be here for that, but my mentor said she would keep me up to date, so.